Thank you, sir. Good morning, everybody. I welcome you on behalf of School of Biosciences. I welcome you all to today's webinar on laboratory diagnosis of infectious diseases, focus on PCR, to be delivered by Professor Ashok Ratan. It's an honor to welcome Professor Ashok Ratan, who is a very senior medical microbiologist of international standing. Professor Ashok Ratan has been earlier part of eminent organizations, including as a faculty member, as a senior faculty member in microbiology at AIIMS, New Delhi. He has also contributed in industrial research in Ranbaxy New Drug Discovery and Fortis Clinical Research Limited. He has also been part of diagnostic labs of Relcare, SRL Diagnostics and Star Metropolis in tertiary care hospitals, including Jain Medical College, AIIMS, and Medanta, the Medicity, and also contributed to public health as a part of CAREC, FAHO, and WHO. Currently, Dr. Ashok Ratan is now with Pathkind Labs as a senior advisor. Professor Ashok Ratan has contributed to more than 100 research publications in peer-reviewed international journals, and he has, to his credit, more than 30 international patents and has also contributed to several book chapters. Today, Professor Ashok Ratan is going to talk on laboratory diagnosis of infectious disease focused on PCR. As we all know, infectious diseases, including COVID-19, has changed the world we live in. The impact has shaken the whole world and is felt more than ever on the biosciences sector. The paradigm shift it brought to the field has emphasized the need for quality manpower in biosciences research and allied fields, and more importantly, in disease diagnosis. In this webinar, renowned international scientist Professor Ashok Ratan is going to talk on a very powerful biotechnology tool, PCR, which is at the heart of modern disease diagnosis. I'm very sure that this talk will enlighten our students and faculty on the impact which PCR has made through the years in disease diagnosis, and more so ever now in COVID diagnosis. So with this brief introduction, I would like to welcome our renowned scientist and eminent speaker, sir. It's over to you now. Thank you, sir.
Thank you, Dr. Kar. Thank you, Dr. Vyas, for giving me this opportunity to interact with the students. Let me start by sharing my slides. So today we will have laboratory diagnosis of infectious disease and focus on PCR. There's a Chinese curse which says that may you live in interesting times. And if you are talking about laboratory diagnosis of infectious disease, you are living in interesting times, because every month, as you'll notice, from different parts of the world, new diseases are either re-emerging or being discovered for the first time. Now, laboratory test itself costs only a little bit of the total medical cost. But even though it may be 2 to 3% of the total cost, the effect or on diagnosis and critical decision making it does is enormous. It's also been seen that once you diagnose a condition, you can find a cure or find a treatment, whether it is with antibiotics, whether it is with cancer, and now with antiviral therapy. There are two types of diagnostic tests. One is in vivo. When you take an X-ray, you have to go yourself to take an X-ray. That is an in vivo test. An in vitro test is what is the laboratory test, where one of the samples are taken from you and they are tested. So you are not required to be present in the laboratory. Those are known as in vitro diagnostic tests. So today we will take you through a journey from 1674, when the first microbe was observed, to 2020, where DNA is the talk of the town. It was one Leeuwenhoek in Holland, who was a draper, and he used to play with lenses. When he put the lenses together, he observed that saliva from his mouth contained something moving, which he called as little animals. And what you see is the first microscope that he had described. Subsequently, this was the time, 1674, that microbiology was born. Subsequently, there was work by uh, Louis Pasteur, who was a chemist, and he was asked to investigate why the French wine was turning sour. And subsequently, there was Robert Koch, who started discovering, he discovered Mycobacterium tuberculosis, he discovered Vibrio cholera, and he has done a lot of research in microbiology. Now, if we cut to what is present, instead of 1882, when Robert Koch was discovering the microbes, you would find that most of the methods that we are using in 2020 are like something that Robert Koch used. Microbiology in 2020 is not very different from microbiology in 1882, which Robert Koch was doing in many of the labs. So if he was to come alive and visit many of the labs, he would have a sense of déjà vu, which actually is a shame. Why manual work is still the norm in microbiology is because people feel that microbiology is too complex to automate. In microbiology, there are multiple types of samples which come in multiple containers, and we have to use multiple methods which only human can handle and machines cannot handle. On the other hand, it's also been argued that microbiology lab is too small for automation, samples are too few for the expenses involved, and the machines are not flexible. However, the ground reality is persons who love microbiology work are decreasing in number. Skill required for isolation difficult bacteria are dwindling, and there are not many labs in India which isolate Helicobacter pylori, Bordetella pertussis, or Yersinia enterocoletica. But the sample load is increasing every year and staff number is remaining the same. The ground reality is also that microbiology results are required 24/7. It's also possible nowadays to do syndromic approach and give detection of simultaneous detection of multiple pathogens. It's possible to take away repetitive, boring work and give a rapid turnaround time, which is known as short TAT. We need to understand the diagnostic cycle. The question will begin, and in the lab, we convert it into pre-analytical phase, where the sample is collected, the analytical phase, where the test is done, and post-analytical phase, where the report is interpreted. Of course, it also involves pre-analytical phase and post-analytical phase. And the TAT is defined as sample from vein to brain of the doctor. The shorter, the better, and that is why automation is required. A word of caution about automation was given by Bill Gates. It said that the first rule of any technology used in a business is that automation applied to an efficient operation will magnify the efficiency. Bill Gates suggests that the second rule is that automation applied to an inefficient operation will magnify the inefficiency. So we must be sure that our processes are right. In my lifetime itself, I have moved from examining urine by a Bunsen burner looking for sugar to examining with a strip using a Eurometer.
That consolidates two industry-proven technologies to perform both urine chemistry and sediment analysis in one small package. This total walk-away system minimizes labor-intensive manual steps and frees operator time for other valuable activities. The easy-to-use analyzer features one-step loading of trusted Ocean dry pad urine chemistry test strips with no calibration required. During analysis, the chemistry module aspirates the sample and precisely dispenses urine on each pad for measurement. The module measures 9 chemistries: glucose, protein, bilirubin, urobilinogen, pH, blood, ketones, nitrites, and leukocytes. For improved accuracy, the color compensation pad accounts for normal urine colors, and abnormal color detection notifies users of false positive reactions. It also performs specific gravity and color and turbidity measurements. The sample rack then advances, based on user-defined criteria, to the flow cytometry module. Here, a probe mixes the sample and aspirates into 2 separate chambers. The sediment chamber is heated to 35 degrees Celsius to remove any amorphous urates. The urine sample is mixed with diluent and fluorescent stain to accurately detect and enumerate formed elements in the sample. It can accurately detect and enumerate red blood cells, white blood cells, epithelial cells, and casts. The analyzer also flags 6 additional elements for review.
So now you see we have moved from manual things to automated things. Overall, if you look at diagnostic tests, you have 2 options. One is to direct demonstrate the offending pathogen. And sometimes you can detect it under the microscope, even an electron microscope for viruses, or you can grow it in culture. Or the other option is that instead of the virus or bacteria or parasite, you can detect its proteins. So the antigen detection is also another methodology available. The other methodology available is indirect evidence. So these are direct evidence are very specific, while indirect evidence will indicate if antibodies are present, then it would mean that in the past you have met with this infection. So for quick diagnosis, direct demonstration is better, but sometimes it is safer and better to do indirect evidence. The test must be specific, sensitive, and precise. And this indicates what is the difference between precise and accurate. Precision is when reproducible results are obtained. That's in the middle slide. But the reproducible results must be as accurate as possible so that there's no bias. And that's what you want from a diagnostic test. In the diagnostic test, you also would determine the sensitivity, the specificity, the positive predictive value, the negative predictive value, reproducibility, accuracy, and for quantitative test, what is the limit of detection and what is the linearity range. Normally, for evaluating any test, you need a gold standard. Gold standard would indicate whether there is disease or there is no disease. And against that, you would find what is true positive and what is true negative, what is false positive and what is false negative. And then you can find out the sensitivity, the specificity, the positive predictive value and the negative predictive value. In the polymerase chain reaction, we would deal with the history first. It was in 1953 that double helix structure was described by Watson and Crick. And in 1962, they were awarded Nobel Prize for this discovery. In 1967, Thomas Brock isolated the bacteria Thermophilus aquaticus, and in 1968, Hargobind Khurana was awarded Nobel Prize for determining the coding structure of the DNA. In 1970, Kleppe, working with Hargobind Khurana, published a paper indicating how to replicate an enzyme replication of DNA using one primer. It was in 1976 that Taq polymerase was first isolated. In 1980, Frederick Sanger was awarded the Nobel Prize for sequencing. 1983, on a moonless night, Carrie Mullins, who was working for CTEX, was driving back home, and he thought that instead of using one primer, we should use two primers. And because Taq polymerase was already available, he combined both the primers with thermostable Taq polymerase, and PCR was born in 1983. In 1985, the paper was published describing the technique, and in 1993, he was awarded the Nobel Prize. Subsequently, we've had the whole bacteria Haemophilus influenzae sequenced, and now, of course, the human genome is also sequenced. And in 2010, RT-PCR methods and protocols was described. This is Mullins, who got the Nobel Prize in 1993, and he was the first one to describe PCR as the way it is. Kleppe, who worked with Hargobind Khurana, and they had published method for enzymatic amplification in 1970, but they were not recognized for their PCR work. In PCR, you require two primers. First, you have to have a template. As you know, DNA normally is double-stranded, but if you heat it to 95 degrees Celsius, it will become single-stranded. Once you heat the DNA to 95 degrees Celsius, it will become single-stranded. Then when you cool it down to 52 or thereabout, then annealing will occur. And so the primers will be able to bind. And a Taq polymerase will be able to add DNTPs and resynthesize whatever is missing. This cycle keeps on occurring, about 30 seconds at 95 degrees Celsius, 30 seconds at 45 to 52 degrees Celsius. Then for elongation, it occurs at 62 degrees Celsius, and subsequently again to 95 degrees Celsius, so that With this cycle, the DNA keeps increasing.
And is a technique for making millions of copies of a gene or a particular DNA target of interest. Copies are made using a very small initial quantity of DNA template, even down to just a single DNA molecule. PCR amplification means that detection and identification of your target DNA can be made using visual techniques based on the size and charge of the piece of DNA you have amplified. The theory behind every PCR, whatever your DNA target, is the same. A mixture is created that contains 5 basic things: your DNA template, a DNA polymerase enzyme, forward and reverse primers and nucleotides, also known as DNTPs, and buffer to stabilize the enzyme. For each new PCR, you choose the primers and design them to be complementary to short sections of DNA next to or near your region of interest. Usually they are between 20 to 30 bases or nucleotides long. The enzyme is there to extend the primers, which have annealed to a specific region of your template, and use the free nucleotides in the mixture to build the new strand complementary to your target. The primers should only anneal to this particular region of interest. During the PCR, the DNA being amplified is heated and the double strands separate. Upon cooling, the primers bind to the template and the process of adding complementary nucleotides by the polymerase can begin. The initial heating step is called denaturation and usually occurs at 94 or 95 degrees centigrade. It needs to be at this temperature long enough to denature the template completely. The following step, when the primers are able to bind to the template at a lower temperature, is called annealing. This temperature depends on the TM, or melting temperature, of your specific primers, and is usually between 50 to 65 degrees centigrade. This step doesn't need to last very long, typically 10 to 30 seconds. The final step in this first amplification cycle is the extension at 72 degrees centigrade. This is the optimum temperature for the DNA polymerase to build the complementary strand, following on from the 3' end of the primer, using the free nucleotides. The polymerase adds them at around 50 to 100 bases per second, so the duration of this step will depend on the length of your target, but 30 seconds to 1 minute is usually long enough for most products. This process of denaturing, annealing and extension is repeated 30 to 40 times, theoretically doubling the number of DNA copies at each cycle, and thereby increasing exponentially the number of copies of your target of interest in the mixture. Samples can be prepared and incubated in a thermal cycler, and a complete PCR reaction can be performed in a few hours, or even less than an hour with certain types of rapid instrument. The lab area used to set up and run your reactions needs to be carefully controlled and maintained. PCR can detect a single molecule of DNA, so it is essential to have a unidirectional workflow with, ideally, 3 separate rooms in your system. Firstly, you must have a clean room. This room would contain all of the reagents for your PCR except the DNA template. None of the reagents and consumables would have been into the area where PCR products are generated or analysed. The best system would involve a workflow where the clean room would not be revisited on a day when you've been in other potentially PCR product contaminated areas. The second room in your system would be the extraction lab, where you've prepared your DNA template ready to put into the PCR. Once you've set up your reaction in your clean room, you would take the reaction tubes or plates to the extraction lab and add your DNA template. From there you can go on to the third room, the PCR machine room. This is where the PCRs take place and where the products are usually analysed by visualisation on a gel or by other methods. It is best to consider that PCR products that you generate contaminate this third laboratory, however careful you are, so never set up a PCR reaction here or take reagents or consumables from this lab into the other rooms, or they could contaminate your clean system.
So this is one way of amplifying. Normally in nature, bacteria takes 20 minutes from 1 to become 2. That is why E. coli from 1 bacteria can become 1 million after overnight incubation. For slow growing bacteria like Mycobacterium tuberculosis, which takes 20 hours or 24 hours to multiply, it takes weeks. But by using PCR and taking 1.5 minutes or 2 minutes per cycle, in 40 cycle, one DNA can be amplified to 1,000,000 DNA. So that small portion which is amplified is known as amplicon. And this amplicon is the one which is detected. So the steps involved are the key steps for an efficient PCR is: one is DNA isolation, second will be primer design, third will be enzyme selection, fourth will be thermocycling, and fifth is amplicon analysis. We have moved from what used to be a manual process of using phenol extraction of the DNA to using spin columns, using the principle that there will be some membranes which will bind to the DNA. And so once we lyse the bacteria or the cell, the DNA will bind to the membrane, then everything will be washed away, and finally the DNA can be eluted in tris buffer. Recently, magnetic beads have been applied, and using the magnetic bead and magnet, extraction of DNA can be automated and can be made high throughput. Primer design is essential, and for that we have to have the sequence that we want to amplify. We then have to make sure that it is of 18 to 20 mer, which is optimal, and that the TM temperature at which it melts is the correct region. Once that is done, then we come to that polymerase enzyme. Traditionally, it is Taq polymerase which is used, but though Taq polymerase is relatively stable, it is not really completely stable. So when PFU polymerase from Pyrococcus fusarius was available, that seemed to be much better, and it has endonuclease or proofreading activity too. But in the recent past, synthetic fusion DNA polymerases have been available, and these fusion high fidelity DNA polymerase have strong proofreading activity, and so the reproducibility is very good, thermostability is good, and amplification is much better than natural enzymes. So it is this fusion high fidelity DNA polymerases which are used whenever you want to sequence something. Type of polymerases are thermoequaticus that we've seen, and then there's also the Wnt enzyme, and what we're looking for is thermostability and high fidelity. After the enzyme has been selected, then 3 temperatures have to be selected. One, as I said, was 95 degrees Celsius, so that where the DNA will be made single-stranded. Then it will be allowed to cool so that the primers can bind. That will normally about 50 degrees Celsius or thereabout. And for increasing, for elongation, you increase it to 72 degrees Celsius so that only real, so that non-specific elongation doesn't occur. And this cycle keeps on occurring. There are many instruments now available. In the past, when they first started, they started with 3 water baths set at 95 degrees Celsius, 45 degrees Celsius, and 72 degrees Celsius. And a person every minute would shift the rack from one to the other. But you can imagine doing this 40 times into 3, that means 120 times in 4 hours time. At that time, it was not too exciting. Now that it can be done automatically, makes it easier. Finally is the analysis. So in the standard analysis, once the amplification has occurred, then it will be analyzed. But that would mean that you run the test, and after the test is over, then you do a gel electrophoresis to see whether amplification has occurred or not. In the final step, a fluorescent dye, ethidium bromide or cyber green can be added. And with that, when exposed to ultraviolet light, you can see whether the DNA is present or not and what is the quantity. Normally, there's a ladder, as the left lane normally is reserved for a ladder, so that you can quantify, you can identify what is the size of the DNA portion that has been amplified. But the standard method gave way to what is now known as real-time PCR. In real-time PCR, the steps remain the same. Sample is collected, the DNA or RNA is extracted. If it is RNA, then one additional step or reverse transcriptase is added, so that from the RNA is converted into cDNA by reverse transcriptase, and thereafter amplification for PCR would occur. And then the advantage of real-time PCR is that you can see the quantity of fluorescence being increased on real time. While the process is still going on, you can see the results. So you don't have to wait for the experiment to be over to see the results. You can analyze the results as activity is going on.
One-step approach and the two-step approach. In two-step RT-PCR, the first step involves the reverse transcription, or RT, of RNA by a reverse transcriptase. This reaction generates the first strand complementary DNA, or cDNA. cDNA is then transferred to a separate reaction tube for the second step of this process. During the second step, the second strand cDNA is generated and amplified by a DNA polymerase. So, in two-step RT-PCR, reverse transcription and PCR occur in separate reaction tubes. This approach is used when there's a need to analyze multiple targets from the same RNA sample in separate reactions, or to store synthesized cDNA for later use. In the other approach, known as one-step RT-PCR, the reverse transcriptase and DNA polymerase are pre-mixed, which allows the RT and PCR steps to be performed in the same reaction tube. Not only is the one-step RT-PCR approach faster, it also requires less pipetting and minimizes possible contamination, thereby improving data reproducibility. It also enables easier processing of large numbers of samples for high-throughput applications.
To begin the reaction, the temperature is raised to 95 degrees. At this temperature, the double-stranded DNA is separated into single strands. Then, three major steps are repeated during each cycle. First, denaturation to obtain single DNA strands. Second, annealing. The temperature is then lowered to approximately 60 degrees. This allows the primers and the fluorescent probe to bind to your target sequence of interest. And third, extension. Between 60 and 72 degrees, the polymerase extends the primers and cleaves the probe, which generates an increase in the fluorescent signal proportional to the amount of amplified product. After one cycle of PCR, there are twice copies of the target DNA. These steps are repeated through around 45 cycles. When the starting material is RNA, one first additional cycle of reverse transcription is required. The real-time PCR reaction requires the following components.
So once you get the results, you would, as I said, you can monitor it in real time. So in the first few cycles, the amount of fluorescence will be low. Once it crosses the detection level, that is known as CT value. That is the threshold cycle, CT, where the number of in which the fluorescent signal crosses the threshold. The probe that is used, so on both sides you have primer, and this primer will bind, and the probe has a fluorescent dye and a quencher. Once that has been that has annealed with the DNA of interest, once that occurs, then the probe as well as the primer will anneal. The primer will use Taq polymerase to synthesize the double-stranded DNA, and as it comes near the probe, it'll cleave that away, releasing the fluorescence. Once fluorescent has become away from the quencher, it will start fluorescing and it can be detected. And this occurs every cycle. And so it will be proportionate to the amount of DNA that has been amplified. If you plot the fluorescence against the number of cycles, then you will get a graph like this, which has 3 stages. First stage is the exponential stage, then there is the linear stage, and finally there's a plateau stage where the reagents are used up, and so it comes to a high level. Ideally, if the DNA has been amplified, it will give you this sigmoid kind of curve.
A new type of coronavirus has emerged from the city of Wuhan in China. Within weeks, it has spread to several countries and has killed multiple people. Corona refers to the virus's distinctive shape. They are a large group common among mammals and birds. But in rare cases like this one, they can spread to humans, a process known as zoonosis. This new virus is the 7th known human coronavirus, and it affects the respiratory system. Some coronaviruses have mild symptoms, however others are much more dangerous. SARS and MERS are both coronaviruses.
So the virus was described in December 2019, and by the 10th of January, the sequence was known, and by the next day, 5 additional sequences were available. This made the designing of RT-PCR. This is an RNA virus, so you'll need a reverse transcriptase, a real-time PCR method, and they found that specificity was residing in the N gene, the S gene is the spike gene, and the envelope gene. And so various permutation and combinations were used to make singleplex or multiplex PCR. When you looked at the biomarkers, it would appear that 1 or 2 days before a person becomes symptomatic, the virus is present in the respiratory tract. Thereafter, the titer increases when a person becomes symptomatic and is maximum around the 3rd day of illness. Subsequently, by the 8th day, the titer decreases. As you notice here in the cartoon, the viral antigen is less than the virus RNA. And in viral RNA, we are going to amplify. So if it is 10, you would amplify to 10 to the power 10. While if viral antigen is present, and antigen detection will detect only the 10 or even less than that. So detecting the virus by RT-PCR will be much more sensitive than detecting the antigen. But viral antigen results can be available in 15 minutes time, as I'll show you, while viral RNA by RT-PCR takes about 24 hours. Subsequently, after 2 weeks of illness, antibodies also appear. So the first week, you will either do RT-PCR, or if you want rapid results, then antigen detection. In the kit that we use from Thermo Fisher, we have a multiplex where a phage, a bacteriophage, an RNA virus, which attacks the bacteria, MS2, is used as a control to find out whether a process is stable to be able to isolate and amplify RNA if it is present. Then we detect 3 virus-specific genes: open reading frame 1AB, the N gene, and the S gene. If one gene is present, then the report is inconclusive. If 2 or more genes are present, then we report as positive. Let's look at this. If you get nothing, that would mean that is an invalid run. For a valid run, at least MS2 must be amplified. If MS2 is amplified, then that's a valid run. But in this case, along with MS2, ORF1A AB is also amplified. But since it is only one viral specific gene, this will go as inconclusive report. On the other hand, if only MS2 was amplified, this will go as a negative report. For a positive report, you will find that at least 2, if not all, the viral specific genes besides MS2 will be amplified. So this will be a positive report. This is again a positive report. Recently, ICMR has also permitted rapid antigen detection kit, in which some material is available in the kit bag itself and others have to be procured. And then using nasopharyngeal swab, the sample is collected. And then in an extraction buffer, the antigen is released so that the virus is inactivated and the antigen, so the virus is disrupted, antigen is released. And then it is, a nipple is put on top of that. And 3 drops are added and through capillary action, if both the control and the test lines light up in 15 to 20 minutes, then it is taken as positive. It was reported from China itself that at best of time, RT-PCR was 70 to 80%. If you add antibodies to it, then diagnosis will be better. But the point is that in the first week of illness, you should do either RT-PCR or antigen detection. And in the second week, if you add antibodies detection, then you will be able to detect more. And this is graphically indicated that RT-PCR is positive in the first week in about 60% of cases. But if you add RT-PCR plus serology, by second week, you will be able to detect all COVID cases. Similar results was also reported by Zhao. The advantage of PCR is that it's easy to develop once the sequence is known, as you've seen. Specificity depends upon the sequence chosen. It takes 4 to 6 hours to get you the results. It can be automated, can be carried out in any adequately equipped lab, and it is the gold standard at this moment for demonstration of the presence of a pathogen. But the sensitivity depends upon the number of factors and is never 100%. Indicates the presence of nucleic acid, but it doesn't mean the presence of living infectious material. RNA is labile and needs special handling, requires specialized equipment and staff, and is costly. So a new test which you would be hearing soon about is loop-mediated isothermal amplification, or LAMP, which has several advantages over PCR, which will be made available, and it is in the pipeline, and I'm sure before the end of the month you will be hearing more about LAMP. The advantages over PCR are it has lower cost and it requires no expensive equipment. It reduces the chances of contamination, low sensitivity to inhibitors, quick results, and results can be observed visibly.
When designing a LAMP experiment, 4 to 6 primers are required, specific to identified regions of target DNA or RNA sequence. Amplification initiates from strand invasion by one of the inner primers, and a strand displacing DNA polymerase extends the primer and separates the target DNA duplex. The first product is then displaced by synthesis, initiating from an outer primer which anneals to an upstream target region. As it is displaced, the end of the product forms a self-hybridizing loop structure due to inclusion of a reverse complementary sequence in the inner primer sequence. This annealing and displacement cycle repeats on the opposite end of the target sequence, and the resulting product is a short dumbbell structure that forms the seed for exponential LAMP amplification. This LAMP dumbbell structure contains multiple sites for initiation of synthesis, from the 3' ends of the open loops and annealing sites for both the inner and loop primers. As amplification proceeds from these multiple sites, the products grow and form long concatenamers, each with more sites for initiation. The result is a rapid accumulation of double-stranded DNA and amplification byproducts that can be detected by a variety of methods.
So the LAMP requires to be done at only one temperature, normally 62 degrees Celsius. So you don't need a thermocycler, though it can be done in a thermocycler, because what you need is constant temperature at one site. And when the results are available, you can see the yellow, which is the color change, acidity because of the presence of DNA. Now, finally, to end the talk, I would like you to have a look at your ability to identify the reaction. Remember that after PCR, real-time PCR, we are looking for the presence of 2 or more RNA sequences, genes from the virus for a positive reaction, MS2 for a negative reaction. So suppose you get this, then your result will be that it's an invalid run. If on the other hand, you get MS2 and ORF1AB, that would mean that it is inconclusive. On the other hand, if only MS2 gene is amplified, it would be that it's a valid test and it's negative. On the other hand, if you get MS2 and all the other 3 viral specific genes, this is a positive test for SARS-CoV. Take-home messages, RT-PCR and PCRs, whether standard RT or digital, are important tools, very useful for diagnosis in infectious disease. It is relatively rapid. They are sensitive and specific, increasingly being used in clinical laboratories. And ICMR yesterday claimed that from one lab, they have increased the capability of detecting COVID in 1,000 laboratories in India. Lab will soon be available, as will sequencing, and every biotechnical student should be comfortable doing this test. I thank you for your attention.
Thank you, Dr. Tan. I think there were a few questions which we have answered online, but still if you have any queries, kindly do write on Q&A. Please, if you have any questions, queries on the presentation or on any laboratory diagnosis, you can kindly raise your queries, ask your questions on Q&A. Dr. Ratan will be happy to answer you. We have 5 minutes more, we'll wait. If there are no questions, I will say thank you to Dr. Ratan and we'll wind up. Some of the questions were very simple, so I answered them, Dr. Ratan, on your behalf. Yeah.